Hello students, welcome to the course BOC 109 Molecular Biology and Genetic Engineering. I am Ms. Marsha Rodriguez, Assistant Professor in Botany, Carmel College of Arts, Science and Commerce for Women, New Goa. The title of this unit is Unit 2, The Structure of DNA and RNA Genetic Material. The name of this module is Structure of DNA Double Helix and Types of DNA. The outline of this module: Introduction, Structure of DNA Double Helix, Chargaff's Rule, Salient Features of Watson and Crick's Model of DNA, and lastly, Types of DNA. The learning outcomes of this module: the student will be able to describe the structure of DNA double helix, the student will be able to explain Chargaff's Rule, and the student will be able to differentiate between the types of DNA. Nucleic acids are of two types, namely DNA, that is deoxyribonucleic acid, and RNA, that is ribonucleic acid. DNA is the chemical basis of heredity and maintains the identity of different species in the world. DNA is organized into genes, which are the fundamental units of genetic information. The structure of DNA double helix was proposed by James Watson and Francis Crick in the year 1953, and they won a Nobel Prize for the same in the year 1962. Two separate and antiparallel chains of nucleotides are wound around each other in a right-handed helical path. Nucleotides is made up of nitrogenous base, sugar and a phosphate group. The sugar-phosphate backbones are on the outer side, and the bases, that is the nitrogenous bases, are paired by hydrogen bonding and stacked on each other. This is an image which represents the DNA double helix. The light blue color strip represents the sugar-phosphate backbone, which is on the outer side, and the inner side you will find the nitrogenous bases. Now the nitrogenous bases are made up of purines and pyrimidines. Purines consist of adenine and guanine, whereas pyrimidines consist of thymine and cytosine. Adenine will bind with thymine and guanine will bind with cytosine. This is an image showing Macklin-McCarty with Francis Crick and James Watson. Now we will learn Chargaff's Rule of DNA composition. Erwin Chargaff in late 1940s quantitatively analyzed the DNA hydrosylates from different species, and he observed that all the species had equal number of adenine and thymine residues. Similarly, there were equal number of guanine and cytosine residues. This is known as Chargaff's rule of molar equivalence between the purines and the pyrimidines. The DNA molecule contains 50% purines and 50% pyrimidines. Now we will learn the salient features of Watson and Crick's model of DNA. The DNA is a right-handed double helix, and it consists of two polynucleotide chains twisted around each other on a common axis. As seen in the image in the previous slide, the two strands are antiparallel, that is, one strand will run in the 5' to 3' direction and the other in the 3' to 5' direction. So they are antiparallel. The diameter of the double helix is 20 angstroms. Each turn of the helix is 34 angstroms with 10 pairs of nucleotides. Each pair is placed at a distance of 3.4 angstroms. Each strand of DNA has a hydrophilic deoxyribosugar sugar phosphate backbone on the outside of the molecule, while the hydrophobic bases, that is the nitrogenous bases, are stacked inside. The two polynucleotide chains are complementary to each other due to base pairing. The two strands are held together by hydrogen bonds formed by complementary bases. That is, adenine will pair with thymine and it has 2 hydrogen bonds, while guanine will pair with cytosine and it has 3 hydrogen bonds. The hydrogen bonds are formed between a purine and a pyrimidine only. In the DNA double helix, you will not find a purine binding with a purine and you will not find a pyrimidine binding with another pyrimidine. It is always a purine which will form hydrogen bonds with a pyrimidine. The complementary base pairing in DNA double helix proves Chargaff's rule. The genetic information resides on one of the 2 strains and it is known as the template strand or the sense strand. The double helix has major grooves and minor grooves along the phosphodiester backbone. Now we will learn about the types of DNA. The different types of DNA are A, B, C, D and Z. Type A DNA has 11 base pairs per turn. Type B DNA has 10 base pairs per turn. Type C DNA has 9.33 base pairs per turn. Type D DNA has 8 base pairs per turn. And type Z DNA has 12 base pairs per turn. Type A, B, C and D DNA are all right-handed, whereas type Z DNA is left-handed. Type A DNA is found under low humidity conditions. Type B DNA is the most prominent form of DNA and is observed under high humidity conditions. And type C DNA is found under greater dehydration conditions. So students, in this module, we have learned the different types of nucleic acid, that is DNA and RNA. We have learned what is Chargaff's rule. We have learned the salient features of Watson and Crick's model of DNA double helix. And lastly, we saw the different types of DNA. These are the references for the module. Thank you, students.